Excellent, thank you. I'd firstly like to thank the organisers for giving me the opportunity to present my work. I recently completed my PhD at the UCL Centre for Cardiovascular and Metabolic Neuroscience, and my work on vagal tone and exercise capacity formed a very large part of my thesis, so I'm very happy to be sharing that with you today. So, markers of parasympathetic activity are well known to correlate with exercise capacity, with elite athletes exhibiting remarkably high markers of vagal tone. Now, it's very much of a chicken and egg story. Either they establish the correlation between the two could suggest that either exercise enhances vagal tone or that there are certain individuals out there that have high vagal tone, either due to genetic or environmental factors that makes them more tolerant of endurance exercise regimes, or it could be a combination of the two. And I'd just like to direct your attention to two interesting papers I found. The first was by Hepburn and colleagues, and this was done in the lab of Professor Coutts, and they showed that respiratory training, independent of any physical exercise, increased the high-frequency component of resting heart rate variability. So this is a marker of parasympathetic tone, and this was complemented by a reduction in the low-frequency component of resting heart rate variability, which is a marker of sympathetic tone. And this was also accompanied by an improvement in heart rate recovery. And what this all shows is that there is some degree, some neural plasticity within the parasympathetic nervous system. There's also some evidence to support the second hypothesis. A study by De Geis and colleagues showed that there was a strong concordance, a very significant correlation between respiratory sinus arrhythmia and exercise capacity, which they derived to be entirely from a genetic factor. And this was done in a twin study, in homozygous twin pairs that were between the ages of 13 and 22. So this experimental animal study was designed to directly test if vagal tone could affect exercise capacity, with the hypothesis that vagal withdrawal could potentially reduce exercise capacity and vagal recruitment could improve exercise capacity. And we did this by targeting the largest population of vagal pregranulonic neurons, known as the dorsal vagal motor nucleus, which we can abbreviate as the DVMN. And the reason why we chose this is that we published a study earlier last year which showed that these neurons play a particular role in mediating the electrophysiological function of ventricular myocytes. And just accepted this year in JPhys, we've shown that a subset of these neurons also have a significant impact on left ventricular contractility. So it made sense then to explore the potential impact of the vagal outflow from these neurons on exercise capacity. So we did this by targeting the DVN neurons because they express a transcriptional factor called FOX2. So using viral vectors with an artificial promoter, PRSX8, we can target these neurons in order for them to express novel proteins. And so to silence these neurons, or to specifically withdraw vagal tone, we transduce them to express the elatostatin receptor from the Drosophila. Now, this is an inhibitory G protein coupled receptor, and on application of the insect peptide ligand elatostatin, you get hyperpolarizing of these neurons, effectively reducing the activity from this nucleus. So this is a very targeted, precise, and well-timed approach to disrupting vagal outflow so that we can ascertain how this affects exercise capacity in conscious animals. And what we found was that at baseline, rats expressing the control transgene GFP and elatostatin receptor had very similar baseline capacities, so there was no significance between the two. But on application of the elatostatin ligand, we saw a dramatic reduction in exercise capacity when these DVN neurons were silenced, and this was noted to recover within a 24-hour period. We then wanted to employ the reverse experimental paradigm. So we wanted to recruit DVN activity to explore the potential impact it could have on exercise capacity. And we did this by transducing the DVN neurons to express a light-sensitive protein, a membrane protein, known as chief. And on illumination via an implanted optic fiber within the brainstem, we can stimulate these neurons to produce precisely timed neuronal activation. And you can see here that we've seen expression both in the nucleus, and when we sectioned a part of the vagus nerve at the cervical level, we saw expression within the nerve itself. And we designed an experimental paradigm. There we go. We designed an experimental paradigm that involved 3 different experimental groups. So we had one group expressing the control transgene, which should not respond to light. a second group expressing the light-sensitive membrane protein, and a third group that was not transduced with any viral construct, but was just classically trained in terms of exercise. So we ascertained their baseline exercise capacity. We then put them through a stimulation protocol. So these two groups received light stimulation for 15 minutes a day for 4 days, and the third group were run every day to the point of exhaustion. And after this 4-day interim period, we repeated the exercise experiments to see how their baseline exercise capacity changed. And in these rats, we also, under isoflurane anesthesia, took high-resolution ultrasound images of their hearts at the end-systolic and end-diastolic time point, so we could derive their ejection fraction and use that as an index of ventricular function. And what we found was that at baseline, all three groups had very, very similar exercise capacities. But following stimulation or exercise training, we saw an improvement in rats where the vagal activity from the DVMN had been recruited, and we also saw a similar and comparable improvement in the group that had run every day to the point of exhaustion. So certainly, vagal recruitment of the DVN could enhance exercise capacity without actually having any physical exercise training. What we also saw was that after the 4-day period of stimulation, at baseline, the rats where DVN activity had been recruited using optogenetics, that their baseline ejection fraction was higher, and to kind of mimic what the heart would be enjoying during exercise, we administered dobutamine, which is a beta-adrenergic receptor agonist, and we found that they maintained a superior response to beta-adrenal receptor stimulation. And I should also add that there was actually no significant difference in terms of heart rate changes at baseline or when given dobutamine. So heart rate wasn't a confounding factor at this point. So we can conclude from this that the DVN does provide essential innervation to the left ventricle, and it plays an important role in determining exercise capacity, which goes hand in hand with our previous works, showing that these neurons play a vital role in mediating the function of ventricular myocytes in terms of electrophysiology and contractility. And lastly, I'd like to thank my main supervisor, Professor Alexander Green, and Professor Sergei Kasparov for providing the genetic constructs, and my collaborators, Dr. Ackland and Marina, for their support. Thank you.